So what my research target? My research target is to develop the drug delivery system against the cancers. Now, if the drug delivery system is successfully developed against the cancer, that drug delivery system can also be used against the other disease or the other abnormal cells which we want to treat. Okay, so this drug delivery system is based on the nanotechnology. Now carbon nanotubules and all are present. Now we need to develop a simple liposome. Okay, now what is the advantage of a liposome is that it will easily fuse with the target cell because of its lipid amphipathic nature. Okay, so it can fuse with the target cell. Now for that we need to mediate that it only binds with the cancer cell, not with the healthy cell. Now this drug delivery system, like this liposome, okay, inside the core we can deliver the toxic substance that will target specifically the cancer cell. Now this toxic substance can be of wide array, like it can carry the classical chemotherapeutic drugs like the doxorubicin or the methotrexate and etc., that in such a way that this drug will only localize in the tumor microenvironment, and the drugs will be directly it will be injected only in the cancer cells and it will not harm the other healthy cells. Okay, so this can be done. Second, instead of the classical chemotherapeutic drugs, we can load the microRNA that will downregulate the gene. Okay, now basically why the cancer is happening, certain proto-oncogenes, they are being highly expressed. Okay, so we need to study for this. We need to, this drug need to be customized. Okay, this drug delivery system and the drugs all need to be customized for the patients. Okay, for different patients, because different patient having a different cancer have different nature. Okay, not every cancer, not every breast cancer, every out of say out of 100 women who are suffering from a breast cancer, now there will be slight difference among everyone. Okay, we need to study that which proto-oncogene is being highly expressed in this woman and which proto-oncogene is expressing in the another woman. Okay, like BRCA1 and BRCA2. Okay, this is the only factor? HER2 receptors are overexpressed over the breast cancer cell. So the drug delivery system that we will develop, it will express only the receptor that will particularly bind with the HER2, and that will facilitate the fusion of that vesicle to the cancer cell only. So this is, so in this drug delivery system we can load the classical chemotherapeutic drug or the shRNA or the microRNA that will downregulate the proto-oncogenes, or we have to, like, we can also send certain genes that can, or certain protein that can upregulate the tumor suppressor gene. Now the tumor suppressor genes in the cancer, like suppose P51 or P53, they are downregulated. The P21, okay, CKIs, okay, CDK inhibitors, they are being downregulated. They must have been downregulated in the cancer cells. So we need to ensure, we need to somehow activate them, okay, by expressing them or by introducing certain, can we send some mRNA, okay, some mRNA, some pre-produced mRNA, okay, means like already a mature mRNA that will go, and we will modify the mRNA that the cancer cell will not recognize it as a foreign, it will recognize that as a self. So it will start expressing those mRNA, and by expressing those mRNA, this cancer cells can be killed. So this can be done. Or what problem is there, that see, because in the cancer is not a we need to think from a multiple dimension. Okay, it's no, the cancer is not occurring due to a one problem or a two problem. There are like the multi-hit model. From multi-hit model we know that there is not one reason of a cancer, or there is not one mutation that is causing a cancer. There are different mutations that have been undertaken by that cell that have caused them the cancer. Okay, so we need to identify them and we need to customize the drug accordingly. That can be done. Now, the second thing also, can we send a cytochrome C? Can we send a cytochrome C, and but it will be particularly targeted to the cancer cells, and the cytochrome C, once if it is introduced inside the cancer cell, it will trigger the intrinsic apoptosis. Can we do that? Next is immune cells. The immune cells is the hot topic, that we need to help our immune cells to recognize those cancer cells. What we can do, that what are the characteristic exceptional feature in that particular patient. Okay, the patient who is being admitted, or the patient which we are focusing on, suppose that patient is having the breast cancer. Now what are the abnormalities, what their cell is exceptionally, what they are expressing, and can we particularly target those cancer cell by using their surface protein, cell surface protein or receptor? Particularly, can we target those cancer cells? And in a cancer cell, the tumor microenvironment is modulated. Okay, the tumor microenvironment, like for example, they start to secrete high amount of hyaluronic acid, so that the enzyme become very stiff, and even the immune cells, normal patrolling cells like macrophages or the T cells, they can't invade the tumor microenvironment. So we can deliver the drug. Drug delivery system can also use to modulate the tumor microenvironment first. That will, like hyaluronidase, that will degrade the hyaluronic acid. That can be done. We can also, like hydrogen peroxide or other superoxides, that can also be loaded inside a drug, and that can be also targeted to the cancer cells. So not only by using the normal chemotherapeutic drug, we can use a wide range of things, wide range of tools, wide range of weapons against the cancer. That weapon can be doxorubicin, methotrexate, siRNA, cytochrome C, like hydrogen peroxide and all. Next, we can also we need to help the immune cells to recognize those cancer cells. Okay, now CD4 cells, CD8 cells, they are not able to invade in the tumor microenvironment, so we can help them to Now another thing that cancer cells do is that they block the immune cells by the immune checkpoint blockade therapy. They used to block the immune cells like CTLA4. They used to suppress the immune cells, and we need to block those CTLA4, particularly expressing over the cancer cells, immune checkpoint blockade therapy. That can be, we can also use that. So we need to target the cancer cells by not a single dimension but by a multiple dimension. And like our defense system of India, like they want to, first of all, they want to do a surgical strike. So they send different teams, alpha team, beta team, gamma team, okay, delta team. The job of different team is different. Same way, we will send different wave of drug delivery system. The first wave, what the first wave will do, the first wave will modulate the tumor microenvironment. The second wave will downregulate the proto-oncogenes. The third wave will be loaded with the hydrogen peroxide, doxorubicin and everything. They will go and target those cancer cells. this can be done. Along with that, okay, so these are the direct weapon which we are using against the cancer. Now beside that, can we, like, what are the important role, what will be the importance of fasting in the cancer? Okay, now recent studies are also showing that different waves, okay, different waves, like, they can also have impact in our body. The fourth and fifth one is the counseling. Like, the patient need to be positive, the patient need to have that courage that yes, I will do it, and I will, that have to be there. And this all process, this need to be very cost effective. This is the most important thing.